shall we get started? So I'd like to welcome Jessica Purcell from the University of California, Riverside, so right around the corner. Usually when people invite seminar speakers, they have like a long history, then they have some anecdotes. But in this case, I actually met Jessica fairly recently, and so I have no anecdotes, but I hope that many years from now I will have, and this will be the beginning of a new relationship here. Local, both due to the proximity spatially and topic-wise. And so Jessica is currently an assistant professor at UC Riverside in the entomology department. She received her BA in biology and history in Williams College.
That's true.
Her PhD in the Department of Zoology at UBC with Lucia Billis, where she did geographic distribution of social systems. She then did a postdoc in ecology and evolutionary biology at Lausanne with Purcell.
Chapuisat,
thank you for helping me pronounce that correctly. On social structure of ants and how elevation gradient influences social systems. And currently her lab works on both proximate and ultimate mechanisms or drivers of sociality. And she does this by looking at both modeling systems and also with field and lab experiments, with population genetic approaches and behavioral approaches and ecological approaches. And so there's something for everyone here in every talk today.
Thanks, Noah, for the introduction and for the invitation. So I'm really happy to be here. I'll tell you a little bit about one branch of my research and maybe a little taster on another branch if there's time at the end. But today I'd like to tell you about the genetic basis of a complex social trait. And I became interested in sociality quite some time ago, and one of the things that really excited me about studying cooperative systems is that we have so many various organisms that show some aspect of cooperation in their life cycle, and the diversity is incredible, yet we don't see sociality or cooperation in the majority of organisms. So I'm really interested in why some organisms are social and others are not, and then how that sociality takes shape in these different systems. So we have everything from very ephemeral social systems to permanent social systems. We have differences in the number of reproductives contributing to the next generation, and we even have some examples where there's probably altruism going on. So some organisms, some individuals are giving up their opportunity to reproduce, even in sort of real time in the dictyostelium slime molds, in favor of others. And so beyond this great diversity of sociality, so this is the obligatory diversity slide, we also know that social organisms have a profound effect on their environment. Partly this is just a numbers game, so we humans are very numerous and we have a profound effect on our environment, but there's also things like caribou migrating through the Arctic, that just because there's so many of them, they are dramatically changing the composition of plants and other organisms where they pass through. But we also have things like naked mole rats and termites that are ecosystem engineers unto themselves. So they're reshaping their environment to suit their own needs, and this in turn affects other organisms around them. And my favorite social organism at the moment anyway, are Formica ants, and they also are ecosystem engineers. So this is a very large wood ant mound from Washington State, and these guys are able to come in and adjust the environment to suit their needs, and I'm really interested in how that kind of plays out and how the underlying social structure of the colony shapes that interaction with the environment. Although that's not really the main focus of the talk today. So when I started to think about really coming down to the basics with social evolution, I started with a Hamilton paper that you may not think of immediately when you think about the evolution of sociality. So thinking about sociality as just a reduction in the dispersal phase. So individuals are more likely to sort of stay together in their natal colony or their natal range, and that may lead to the conditions necessary to initiate cooperation. So this Hamilton and May paper from 1977 explored theoretically the tension between the pressure to emigrate or disperse and the pressure to remain philopatric or stay in the natal range, and really boiled this down to the trade-off between dispersal risks and local competition. And so thinking about a few of these risks, I'm going to come out here and keep wandering around. So on the dispersal side, obviously if you're going out into a new environment, you may face unknown risks such as encountering more predators. You may end up in an environment that is already saturated with conspecifics, or you might encounter new competitors. And you may get somewhere and realize that all of the females have already been taken, or if you're a male, or if you're a female, all the males have already been taken, and well, you're just out of luck. But at the same time, if you choose to stay in your natal environment, you face local competition with others who may be more likely to be kin, and this can be competition for resources, can be competition for mates, but then you also face the potential for crowding, and crowding can lead to other kinds of problems, such as the increased prevalence of disease or pathogens. And so this trade-off kind of intrigues me, and I like to come back to this when I'm thinking about transitions in social structure. Of course, the more famous Hamilton equation is the inclusive fitness theory, where he's thinking about the role of relatedness as well as costs and benefits of cooperation in driving these transitions. And I often put up this slide for many audiences because I like to try to convince people that little r is not the only part of this equation. A lot of people kind of get stuck on little r because it's easy to measure. I don't think I need to convince you guys of this. This is a slightly different audience than I'm used to, so that's great. But I want to kind of emphasize where the costs and benefits in this inequality can come into play. And so thinking about this in terms of the trade-off between emigration and philopatry, if we think of it as a balance, what favors the emergence of tolerance and cooperation? And I'll actually broaden out that comparison a little bit to talk about dispersal versus joining. So dispersal in terms of an individual going out and founding a new nest independently, or potentially remaining in their natal patch, or relatively local and joining an existing nest. So they're the similar kinds of costs and benefits associated with them, but perhaps a bit more general. And I'm going to put it into this context because, as Noah mentioned, I'm interested in transitions in social organization, and the one I'll be focusing on today is a transition from single queen colonies. So these are colonies composed of a single queen and her offspring, or in other words, close relatives, to multiple queen colonies, where you often have non-related individuals working together, reproducing together, and building a colony. And this transition has occurred in my favorite genus, Formica, as well as in other groups of ants, as I'll show you in a few minutes. So if we think about this transition to groups composed of non-relatives, what are some of the factors that could start to shift the balance towards it being beneficial to join an existing group of individuals, even if they're not related to you? So one of the obvious ones is if dispersal or independent founding is particularly risky in the environment that you find yourself in, it might be a good idea to join an existing colony. It might be a good strategy. So this is an interesting one. Another one that I've explored, and I was just talking with Julie about earlier, is the potential that there might be synergistic benefits to cooperating with other individuals. And so my favorite example of this is from my social spider work during my PhD, where groups of spiders are actually able to work collectively to overcome much larger prey items than they themselves are. And if you compare this to solitary living spiders, the spiders are generally able to capture prey that are much smaller than they themselves, but by working together they can get these much larger resources. So that's a good example of a synergistic benefit. But I also put this in. I've done a little bit of theoretical work on it, but also as a way to shamelessly promote some work that I won't have time to talk much about. So I've done some fun stuff on ant rafts. And this is another, I think, synergistic benefit that can emerge from forming a group or staying and joining a group. And these groups don't have to be related. So these are some examples of ant rafts that you may have seen in the news from a year ago during Hurricane Harvey. So unfortunately, these are fire ants, so you definitely don't want to poke at these guys. But what happens when their environment floods during a hurricane or similar is that they just kind of come together and form this complex collective structure, and they can float, they can remain afloat for multiple days. If you put an individual fire ant in water, it will sink. And so I've worked on this, but in a much friendlier genus, so one that doesn't sting. Turns out that fire ants actually increase the amount of venom in their stings while rafting, so you really don't want to poke a fire ant raft. But looking at this in Formica, which actually don't sting, so they're much nicer, we designed a setup where we were able to place our workers on or our colony on a glass stage, and we could film from above and below. We could raise the water level slowly with an IV dripper, and we asked a bunch of questions about whether colony members are exhibiting altruism during the formation of rafts, or some individuals giving up their lives for the good of the colony. And was there a consistent division of labor during raft formation? And because this is a shameless aside, I won't tell you about all of our results, but I will show you a video. So this is one example of a raft forming in our lab setup. We're filming from below here, and so we have some workers painted. These are sexual larvae, so you can imagine that these are pretty high value individuals in the nest. These are going to become gines or males. And again, we're filming from below, and the water level is rising gradually, so you can see that it's somewhat accelerated. The workers are kind of freaking out, and they're jumping on board those larvae. And the larvae make up the raft. So these workers are actually floating on not only their babies, but their high-value babies, the ones that will be the future reproductives. So this was an initially puzzling result, and it was a lot of fun to play with. Turns out that the brood are very buoyant. They're much more buoyant than the workers themselves. So they're taking advantage of that property of the brood, and it's also not killing them. So the brood can remain submerged for hours, as can workers actually, and still come out unscathed. So this does not cost these brood their lives, at least in our very controlled lab setting. So rafting's cool. Okay, back to the rest of the talk. All right, so what I'd like to talk about today is a different, less of an ecological and more of a genetic driver that may tip the balance towards joining and away from this dispersal phase. So this is using selfish genetic elements. And the key here, and I'll walk you through my logic, is that individuals don't have to be related to each other across most of the genome if they share a selfish allele. And so the thinking about this dates back to Richard Dawkins' The Selfish Gene, where he built upon Hamilton's rule to promote the idea that selfish genes may exist and promote their own transmission at the expense of other parts of the genome, or even at the expense of the whole organism. It doesn't seem like a very social thing to do. So how does that link in with sociality? So in theory, this can actually lead to cooperation among bearers of the selfish allele, and I'll show you how that might work. So Dawkins proposed this analogy with the greenbeard, and I would have loved to give you some ants with greenbeards, but it looked like they were vomiting, so I gave them green hairdos instead. Okay, so the most famous empirical example of this greenbeard was found in the fire ant actually 20 years ago. And again, these are stinging jerks, so I really like my Formica, they don't sting. But the idea here is that the individuals that bear the greenbeard allele can recognize other individuals that also bear the greenbeard allele, and they preferentially direct cooperation towards those individuals. They then don't cooperate, or they can actively aggress individuals that lack that greenbeard allele. And that's in fact what they found in fire ants, and I'll come back to that in a little while. But I also want to give you the idea that this isn't the only type of segregation distortion mechanism or selfish gene mechanism that could lead to cooperation. So there may be other types of segregation distortion that could have a similar outcome. So today what I'd like to do is start off by telling you about the transition that I'm interested in, so that transition from monogynous to polygynous, or single queen to multiple queen colonies in ants. I'll then tell you a lot about what we know about the genetic basis of that transition, and hopefully with a little bit of time, just a little bit about what we know about the environmental drivers, although that's a major branch of my research. And then I'd like to end by trying to link together these two ideas, which is something that needs more attention in the field, in my opinion. But before I go too far, I want to acknowledge that this is for sure not just my work. So I have lots of collaborators from my postdoctoral institution, the University of Lausanne in Switzerland, as well as others who are in other parts of Europe, other European institutions, my lab here at UC Riverside and my collaborators. And we also have generous funding from a number of sources. I also want to make sure to acknowledge the excellent photographers. So I'm not a very good photographer, and I've shamelessly used wonderful photographs by Jean Meunier, Daniel Pierce, and Alex Wild. I tried to acknowledge them, but just in case I forget, here you go. All right, so let's go to the Alps. This is actually the distribution of my focal species for the next little bit of the talk. So very conveniently, it's located in a beautiful place. Love working there. And Our protagonist is Formica selysi. So this is a subterranean ant, tends aphids, collects protein opportunistically, and it's a floodplain specialist. So this is what you saw rafting before. It lives in these nice mountain floodplains, you know, preferably with wine regions just above them. I really love field work. And what's really especially neat about this species is that we had, when I started my postdoc, already a long-term study population that had a good mix of monogyne and polygyne colonies within one single population. So this is a pretty big advantage over even some of the other polymorphic species that have been studied, because in many species there will be a monogynous population here and a polygynous population over here. But in this case we have both forms living in the same population, right, in sympetry. So this allows for a lot more comparative analyses. So the Formica social system, this is a bit of the state of the system when I entered it. So I contributed to this, but so back in 2010 when I started my postdoc, we had no evidence of divergence between the two social forms in this place based on a microsatellite loci. So there were 8 polymorphic microsatellite loci that had been used across all of the Formica that have been studied. The polymorphism was found to be stable for 15 plus years in our focal population, and this included over the course of colony deaths and turnovers. And then the colonies were not, and still are not, geographically clustered by social organization. In fact, we can't find strong evidence of microclimatic or microhabitat factors that shape their distribution in this place. Then looking at the social polymorphism, so if we compare the single queen colonies to the multiple queen colonies, there's actually a suite of related differences. So for example, in the single queen colonies, queens and worker body sizes tend to be larger than the multiple queen colonies. The queens are more fecund, they live longer, but the colonies survive for less time because they don't requeen. And then there's some evidence that there's differences in the founding tendencies. So the polygynous individuals tend to be more dependent founders, they tend to join existing nests. And this syndrome, this polygyne syndrome, goes beyond the Formica system that we were looking at. This tends to be consistent across other socially polymorphic ants as well. All right, and I keep alluding to these other socially polymorphic ants. So this is kind of a very tiny slide, but I'll walk you through the take-home message. So the current best thinking about in the field of ant evolution is that the common ancestor of all ants was supposed to be monogyne and monandrous. And then if we look across the phylogeny, so this is just some of the most common genera, we see that polygyny, or this presence of multiple queens in a nest, has evolved multiple times. These are the blue lines. And then the dotted lines show genera where there's actually a social polymorphism. So either there's closely related species that differ in social structure, or we can even find both monogynous and polygynous forms within the same species. So this is something that's evolved repeatedly. And as mentioned, it can be a little bit hard to explain initially because these are groups of non-relatives. All right, so I'd like to tell you about our work investigating the genetic basis of this transition. And this started more than 6 years ago now, where we really didn't think we would find anything, to be honest, because we hadn't seen differences in microsatellite loci. But we decided to try this newfangled rad sequencing. At the time, it was newfangled. So I'm calling it a bunch of different things because it's not really the published version of rad sequencing, nor is it the published version of genotyping by sequencing, but it's essentially like double digest rad sequencing. I'm happy to share our protocol. It works very well with tiny amounts of DNA. We also paired that with some whole genome sequencing, but at the time, our genome assembly was fairly poor. For our initial study, we did an association study, so we used males, which are haploid in ants. We did genotyping by sequencing on monogynous and polygynous individuals. We then constructed linkage maps, high-density linkage maps using rad loci. So using offspring of a single queen, but we actually produced a lot of linkage maps, as you'll see. And then this Formica didn't have a genome, so we produced a draft genome, which at the time was okay. We've now since assembled one pretty much to chromosome. All right, so to show you the results of this first study, I'm going to show you a rather complicated figure. Don't panic, I'll walk you through it. So this is part of a Circos plot. These green bars represent the linkage groups from our high-density linkage map. We recovered 27 linkage groups, which matches the expected karyotype of Formica. So it should have 27 chromosomes for a haploid, and we found 27 linkage groups. Good sign. On the interior part of the circle, we've mapped our genomic scaffolds. And because these are relatively small, you can see how poor quality our genome was, but it was still much better than just having the anonymous rad markers. And then on the outer part of the circle is the interesting information. So this is differentiation between the single queen and single queen and multiple queen individuals, and so plotted as a fixation index. So the high values represent positions in the genome that are highly differentiated between the two forms, and the bright red dots represent points that are highly differentiated, even with a very, very conservative false discovery rate. And so what you can see clearly is that these are clustered on one chromosome, but they span most of this chromosome. So it's not just one region, but it's a large portion of this chromosome that's highly differentiated between the two forms. So this is a signature of a supergene, and indeed this region harbors more than 600 different genes. So this raises some immediate questions, including how is such a large region being maintained at such a high FST value? So across this region, there's a lot of points that have an FST of 1, so they're perfectly differentiated between the two forms. And to address this question, we produced more linkage maps. So this is a plot showing linkage maps of monogynous individuals. So here and from now on, I'll be referring to the monogyne associated haplotype as M or SM, but usually M because I'm lazy. And then the polygyne associated haplotype is P. So we're comparing an MM homozygote linkage map to MP heterozygote linkage map. And this is actually combined 4 different linkage maps. And what we find is that this whole region with high differentiation between the two haplotypes has 0 recombination in the heterozygotes. So this means that this is a huge non-recombining region. And that's the mechanism by which all of this differentiation is being maintained across such a large region. We then went on, and this is still unpublished, we then went on to produce PP homozygote linkage maps to look at what actual physical differences were maintaining this non-recombining region. And this is a bit of a mess, but you can see that there's at least 3 inversions and other translocations. So it's the shuffling of the order of genes that's maintaining this non-recombining region. And I'll come back to this in a little while. So far we've been focusing on what's going on in Formica, and it turns out that another species has been relatively well studied for a similar trait, and that's Solenopsis. So these are two independent origins of polygyny and of a polymorphism in social structure. And we then went on to compare our supergene to the supergene that was discovered around the same time or a little bit earlier in the fire ant. And so this now I've added to the circos plot. This is the linkage map of the fire ant. These are their genome scaffolds, and you can see that it was better assembled at that time than ours was. And then the blue lines represent the positions of their supergene, which is chromosome 16, in the Formica selysi genome. And the green lines represent the position of the Formica selysi supergene in the fire ant genome. And you can see that there's no gene content overlap. So even at this point, we could see that at least if there was any gene content overlap, there wasn't a lot. And now that we've produced better genomes, we know that there's absolutely no gene content overlap. So this was really astonishing, because these fire ants, they have the same polymorphism, they have the same polygyne syndrome, and they also have a supergene, but these supergenes are independently evolved and non-overlapping. So then we could also compare other characteristics of the way that the genotypes are assorted into the colonies between the two species. So looking first at Formica selysi, now our monogynous colonies completely lack the P haplotype. So all of the individuals, the queens are homozygous MM. They produce hemizygous M males and homozygous MM queens and workers. They never, at least in the mature colonies that we have looked at, they never mate with P males. Whereas in the polygynous colonies, we have heterozygous queens and homozygous PP queens. And these are producing heterozygous and PP workers. However, we know that the MP queens can mate with M males yet we never see individuals in these polygynous societies, and I'll come back to that in a moment. But first, let's contrast that with the fire ant case. So for historical reasons, because the supergene was initially discovered as an allozyme in fire ants, we have two haplotypes. These are called big B and little b. So the big B haplotype is always homozygous in the monogynous colonies, just like in Formica selysi. In the polygynous colonies, all of the queens are heterozygous, and in fact, this is where the greenbeard acts. So there are both heterozygous and homozygous big B workers in the polygynous colonies of fire ants, but the heterozygous workers will actually kill any joining big B queens. So this is one of the mechanisms by which this polymorphism is maintained. The other mechanism is that the little b haplotype, these guys can survive to adulthood, both queens and workers, but they're never found in colonies. So they're never found reproductive. So this is essentially a homozygous lethal. And so it's fairly easy to understand how this polymorphism is maintained. But it's a lot harder to see why these two social forms aren't just diverging. They're not really dependent. You could have just PP colonies forming the polygynous societies and just PP queens and MM queens. So why is that sort of connection being maintained? And this is something that we're actively working on. So I'll give you a little sneak peek. Oh, the other thing I wanted to mention here is that because we don't have any sort of lethality in our PP homozygotes, there's no evidence right now of P degeneration in Formica. And this is fairly unusual in other supergenes, to have both homozygotes being successful and reproducing. So one of our first questions was, all right, so we find these heterozygous queens mating with M workers in the field. So why aren't we finding any MM queens or workers in these polygynous colonies? What's happening to these guys? Why aren't they being produced? And so this is the point where we can start to ask whether the Formica supergene is selfish. So what's happening to these individuals? And I'll walk you through some of our ongoing work, but this is work that's being carried out by Amaury Avril, who just finished his PhD at the University of Lausanne. So I won't go into as much detail because it's kind of his story to tell, but I'll give you the punchline because it's important for the rest of the story. So he asked, is there biased transmission of the Formica supergene? And he had a series of hypotheses for what kind of bias could exist. So one idea was that there would be meiotic drive, and this is the biased production of gametes. So this is an example from the stock-eyed fly. If you have a heterozygous male, all of his sperm are going to have the big D allele because the little d allele just fails to produce viable sperm. So this is sort of an example of meiotic drive. It's a relatively common segregation distorter in different systems. So he tested for this by essentially having many offspring of heterozygous queens and looking for any kind of transmission bias, and he didn't find any. So they were right on the 50-50 expectation. He then looked for the presence of a greenbeard allele. So did he see differences in the behavior of the individuals bearing the P haplotype towards others that either had or did not have the P haplotype? And again, he didn't find any evidence of this, neither neglect nor active aggression towards individuals that lack that allele. And he did this in a number of contexts, both with developing brood and with queens. Finally, he looked for developmental incompatibilities. So this is sort of developmental arrest of certain offspring during development. So he had both field crosses with heterozygous queens crossed with M males, and he also did some additional ones. So he genotyped nearly 400 eggs from these crosses and 230 larvae. And this is a little bit small, I apologize, but this is the proportion of heterozygous individuals on the y-axis. So at the egg stage, he had the expected, and I'll remind you that ant males are hemizygous, so they're haploid. So our expectation from this cross would be 50% MM individuals and 50% MP individuals, and indeed that's what he found with the eggs. So 50% were heterozygotes, 50% were MM. But when he looked at the larvae, 100% of the larvae are now heterozygotes. So what's happening is that the eggs are simply failing to hatch. The MM eggs, the offspring of heterozygous queens, are failing to hatch. And he did a number of tests to ensure that MM eggs aren't just generally bad. So if they're the offspring of an MM queen, they're just fine. But it's just in this cross. So there's something about the mother having the P haplotype that's really bad for MM offspring. And to summarize this part, I've showed you that there's a supergene underlying queen number polymorphism in Formica selysi, that the P haplotype is acting as a selfish gene, so the offspring of these heterozygous queens that lack the P haplotype just fail to develop. And it turns out that this segregation distortion mechanism does exist in some other systems. It's sometimes referred to as a maternal effects killer, and it resembles the Medea system in flower beetles, where there's actually a cytoplasmic toxin that can be rescued by one of the haplotypes if the mother bears that haplotype, and then, or sorry, if the offspring bears that haplotype, and then also a couple of systems that have been described in C. elegans. But this is the first description that we know of in a social insect, so this is pretty exciting. So I'll switch gears and broaden out our scope a little bit for the next section and ask whether the Formica supergene is actually widespread. And Formica as a genus is a bit of a mess. It's found in North America, Asia, and Europe, and the best phylogeny that was published this year is just focused on European species. But I was also focused on European species at the time, so this is pretty useful. So I've been telling you about what's going on in Formica selysi, but now we're going to broaden our scope and look at a number of other species. So there's actually 2 other Cerviformica, but only one is shown here. We're also going to look at another subgenus, the Coptoformica, and the Formica exacta is the species, and then Formica truncorum. And these are all species that have a described phenotypic polymorphism in queen number. So do they have the same supergene, and if so, what's going on with that? So here's some nice pictures. They're very attractive ants. I think everyone should work on them. They don't sting, they're lovely. So in order to study this, we used RAD sequencing and paired with whole genome resequencing of both monogynous and polygynous individuals. I'll show you some of our data. So linking it in with the phylogeny, you've already heard a lot about Formica selysi, but I'm going to show you this figure to get you on the same page with how we're going to be looking at this data. So this is now a principal component analysis of RAD sequencing data from just the supergene, so just that region of differentiation and suppressed recombination. And what you can see is that these are the individuals of monogynous origin, these are the individuals of polygynous origin, these are all males, and you can see that PC1 nicely separates the two. So if we do the same thing for Formica truncorum, again, we just have males. We see the same pattern. So the individuals of monogen origin are here, polygen origin here. We have 2 haplotypes. They're associated with social structure. And here's Formica exacta, and this time we actually have workers. So it turns out that this is a PP homozygote, these are heterozygotes, and then these are MM homozygotes. And again, it's perfectly associated with social structure. So it looks like our supergene is older, relatively old, and conserved across these different subgenera, at least in Europe. So again, there are distinct supergene haplotypes in each species. I'm not showing you a couple of cases where it's actually a little bit messier, and these are in the same subgenus as Formica selysi, but I'll come back to that towards the end of the talk. And these haplotypes cluster by queen number phenotype. So they generally cluster. It's not perfect in some species, but generally they do. So then we have this raw material to investigate how the supergene actually evolved across the genus. And this is a nice opportunity because we don't know a whole lot yet about how supergenes evolve, although we keep discovering new ones. So these are pretty important structures in a lot of different organisms. And the a priori prediction across many different supergene studies is that they should develop like sex chromosomes, because sex chromosomes are the best studied case of a supergene. And the way that sex chromosomes develop, the other reason that we thought that this would be the case is because we found these multiple inversions, which looks kind of like the expanding strata model for sex chromosome evolution, which I'll walk you through now. Okay, so in the expanding strata model, which has been actually observed in mammalian and avian sex chromosomes by these guys initially and then a lot more following up on them, is that recombination halts in one region of one chromosome. And this is a very simple overview. So both variants persist through speciation events. And then here's the important part, why it's called the expanding strata model. So this non-recombining region expands in, say, one lineage. So I'm showing that with the light blue here. So perhaps there's a second inversion. So recombination may have ceased initially through an inversion. There may be a second overlapping or adjacent inversion. And this can keep continuing. And so we can have this sort of growing non-recombining region. And of course, later on, we get degeneration of the Y, or the W haplotypes. And the way that this would work, if we look at differentiation between, so now putting into the terms of the supergene, between M and P or between males and females across different species, is that we have this initial inversion. This raises differentiation across that entire region. As that inversion persists, that differentiation is elevated relative to the sort of ongoing recombination at the ends. And if we have a new inversion, it elevates as well, but not as much. And so we have this sort of march on towards the oldest strata, or the initial inversion, having the highest differentiation between males and females, or between M and P haplotypes in our case. And this results ultimately in two extremes. So we have the young strata topology, where individuals with the M and P haplotype cluster by species in areas where recombination is ongoing. And we have the old strata topology, where all of the M haplotypes and all of the P haplotypes cluster even across very divergent species in areas that recombination has been suppressed for the entire evolutionary history of differentiation. So now onto our data. Doesn't look like that. So this is a saguaro plot, and I'm going to walk you through what it means. So this is a relatively recent innovation in how to analyze differences in topology in genome sequencing data. So this is now just looking at the position along our supergene. These blue regions are the recombining ends of the chromosome. All this gray stuff is in that non-recombining region. And Saguaro uses a hidden Markov model to identify tree topologies in sliding windows, and basically to identify natural breakpoints in those topologies. So here I've highlighted in blue that young strata topology. So this is where individuals are clustering by species. And then in red, these little tiny red notches are areas where we have the old strata topology. So this is where individuals are clustering by social form and not by species. So here we have actually trans-species SNPs. And the gray areas are sort of intermediate topologies, but I won't go into those today. So these tiny little red regions harbor trans-species SNPs, and these regions, interestingly, span the entire non-recombining zone of the supergene. So what we were predicting with the expanding strata model is that maybe one of the three or more inversions would have a large red region, right? So a large old strata associated with it, but instead we have these little tiny broken up strata. And just to show you this in a way that might be a little bit more familiar, so this is now looking at a fixation index, but it's a trans-species fixation index, so it's a little bit hard to grasp at first. But basically, the high up, the values that are relatively high, and especially the ones that are red, represent regions where the M and P haplotypes are differentiated across all of the different species. And in fact, the red dots are 1 kb windows that harbor trans-species SNPs. And so these are lining up very well with our saguaro red topologies as well. So what this tells us so far is that we have very small regions, and these regions may be very ancient because they have these conserved trans-species SNPs across quite ancient divergence. We then added in additional species, and this is a little bit of a nightmare to look at, but we have 6 individuals that are from monogynous colonies. These are from different species. We have 4 from polygynous colonies, 3 of which are heterozygous. The blue boxes represent individuals that have the monogyne associated haplotype, what we would expect from, and these are just focusing in on these tiny regions with trans-species SNPs. The red represent individuals that are homozygous for the P haplotype, and then the little triangles are heterozygotes. And so using this approach, we were able to further narrow down a very small region that appears to have trans-species SNPs conserved across 15 different species. And using this approach, and this is something that's been done in sex chromosome evolution as well, we can really narrow down our set of candidate genes that are actually underlying the polymorphism, the phenotypic polymorphism that we're interested in. The other point that I wanted to make, so now I'm plotting our saguaro plot is here in the middle. This is actually a monogyne linkage map from another species, Formica exacta. It's very collinear with our monogyne genome from Formica selysi. And then these are the inversions from the Formica selysi PP against the Formica selysi MM. And one thing that was really striking, so again, we expected sort of one inversion to maybe have a large section of old strata. That's not what we found. We also didn't find all of these little red sections to be found right at the inversion breakpoints. So it doesn't seem to just be a structural constraint. There's also potentially some balancing selection going on. To explain this pattern, we devised a new model. So we call it the eroding strata model for supergene evolution. And here it starts off very much like the expanding strata model for sex chromosome evolution, where we have recombination halting in one region of the chromosome due to something like an inversion. Both variants are persisting through speciation events. But then here's the difference. So rather than having new inversions forming, we propose that there could be rare recombination events between these two haplotypes, probably centered towards the center of these different inversions, that are homogenizing small regions within lineages. And then if this keeps repeating, we might end up with something that looks more like this. So here again, we're looking at differentiation between M and P across species, but we're ending up with this much more broken up pattern where the eroding red strata is getting smaller and smaller due to these very rare recombination events that are fixing in different lineages. So to summarize from this section of the talk, we found that the supergene in Formica seems to be an old polymorphism, an old genetic polymorphism. We have very small regions of the supergene harboring SNPs that are shared between social forms in 15 different species. And the genes in these areas are excellent candidates for further investigation. And in fact, we've been doing that investigation, and we find social form specific expression in these genes, or biased expression in these genes. So we're interested in pursuing that further. And this supergene doesn't exhibit the standard strata like sex chromosomes, so we don't see a signature of an expanding strata. Rather, we propose an eroding strata model for supergene evolution. And the last few things I'll say about supergene is to ask, okay, if we broaden our scope even further, so we've been focusing on Europe, and we've been just focusing on one individual, to ask how the supergene is actually functioning. So if we look more broadly, does it always work the same way in different critters, and also is it present in North America at all? So in order to get at this, so I moved to UC Riverside 3 years ago, and immediately decided to haze my lab group by bringing them on a massive field trip. So we collected from Riverside all the way up to the Arctic and Alaska along 2 latitudinal transects. We also collected along 3 major elevation gradients, and so we got over 10,000 individuals, definitely well more than 50 species. We've sequenced more than 6,000 of these so far with RAD sequencing. And you can't read this at all, doesn't matter. The point is that there's way more diversity present in North America than there was in Europe. So we've been focusing up until now on these little boxes, and now we have a lot more Formica diversity to work with. And this is a collaboration between my lab and that of Christian Rabeling at ASU. So we've now started to look at some of our widespread species. So this is an example of Formica neoclara, which is distributed in California and also in Canada. So if we just compare genome-wide differentiation, we recover very clear signatures of sort of spatial structure. But then if we look at our supergene, we find that there is some interesting variation spatially. So this is the supergene in California, supergene in Canada. In California, we didn't find any phenotypically monogenous colonies, but we did see some structuring at the supergene. But in Canada, we did find some variation. And what's cool here is that these individuals are heterozygous across the whole length of the supergene. So I think we have 2 haplotypes here, but there's imperfect association with phenotypic social structure and really distinctive regional variation. So scaling out to all of the species we've been able to look at so far, both in North America and in Europe, we have a bunch of different things going on at the supergene, and this is really intriguing. So we have a number of species that seem to have this selysi-like distribution that I showed you before, so maybe they have a similar maternal effect killer going on. We have some species here in North America that are Invicta-like in that they lack a PP homozygote. We have several different groups that have 3 haplotypes at the supergene, and we have some that have regional variation or are otherwise variable. And so there's a lot of interesting variants to work with, and I love working with natural variation. And in fact, our proposal to study these 3 haplotype systems was just funded by NSF, so we're excited to move forward with this. But for now, what we know is that there are different genotype distributions, and at least in my mind, this suggests that the mechanism maintaining this polymorphism may differ across different species. So on that note, I'd like to take just a few minutes to tell you a little bit about the whole other branch of my research that I'm excited about, but didn't want to bore you with too much today. So looking at environmental variation and how that contributes to this transition. And I promise it will be brief. So I ask a bunch of different questions. Some of them include whether monogynous and polygynous societies differ along biogeographic gradients. So do they differ in their distributions? Do the social forms differ in their response to extrinsic factors? And today I'll just talk about the patterns, so these biogeographic patterns, the first question. I combine different approaches, so I do field observations, I do these big surveys like I showed you a moment ago. I also do lab manipulations. Again, I won't tell you about the lab manipulations, but feel free to ask if you're interested. I'll tell you about some field observations. And we'll start back in the Alps with Formica selysi. So this is work that I did in collaboration with Loic Pellissier, who's now at ETH Zurich, but at the time he was a graduate student, and with Michel Chapuisat. And I sampled populations of Formica selysi. So we'd been working in one single population in the Rhone Valley here for many years. But I sampled 17 additional populations across elevation gradients and in different drainage basins in France, Italy, and Switzerland. And I was interested in what the genetic structure of the populations was, whether there were consistent shifts in queen number along elevation gradients, and whether there was a loss of polymorphism. And we found that there was a very strong structuring between different valleys. So this is just a haplotype network summarizing the variation that we saw in the mitochondrial sequences. So just to remind you, or if you haven't seen a haplotype network lately, all of the circles represent a single haplotype. So the larger the circle, the more individuals share that haplotype. The little bars between the circles represent one base pair difference, and any hashes represent additional base pair differences. And so we found nearly perfect association in the haplotypes between these different valleys. And I want to point out the Rhone drains to the Mediterranean, the Po drains to the Adriatic, and the Rhine drains to the North Sea. So these are quite isolated, even though they're relatively proximate in geographic space. So this set a nice stage for within valley comparisons of the distribution of queen number variation. And in fact, we looked at the proportion of monogynous colonies summarized across all of our different samples in these different drainages. And we found a very consistent pattern of increasing proportion of monogynous colonies at higher elevation. So this means that each of the social forms, in addition to having this big genetic region differentiated, they also are experiencing different average environments. So one of the things that I'm interested in is why they're not speciating. And I guess that will come back to the selfish gene and some other things, but this is cool. We were able to take it a step further to identify, using GIS, abiotic factors that were associated with social structure variation. And we found that the temperature, particularly the temperature of the worm quarter and season length, were highly associated with the presence of polygynous, or the frequency of polygynous colonies, but precipitation and flood rate were not. And so this allowed us to then go on and experimentally manipulate temperature and look for things with that. But to summarize this section, we found that polygyny may be advantageous in some environments. So for example, in warmer environments or those with longer growing seasons, and monogynous may be advantageous in others, although we're not totally sure whether this is a selective thing or just sort of a passive sourcing dynamic. I just wanted to make the point that these biogeographic patterns are really useful because they can inform predictions about environmental factors that are shaping a focal trait, and they should always be paired with follow-up experiments. So this is very much a correlation, and then it's important to take the next step to actually do some experiments, which I did, but I won't talk about. Okay, and then with this nice big sample from this big latitudinal gradient, what more can we learn? We can look for intra and interspecific patterns in the distribution of social organization in North America, all the way from the high Arctic down to the deserts around Riverside. And we've started to do that just looking very preliminarily at some of our most widespread and abundant species. So here I've just plotted the approximate latitudinal distribution of these 6 species, and then the frequency of monogynous and polygynous colonies along the latitudinal gradient. And actually in 5 out of the 6 we have the same predicted pattern that we saw in the Alps, except now with latitude instead of elevation. So more monogynous colonies at higher latitudes. And the reason I show this is this doesn't look like it's significant, but in fact it is. It's just that Formica frankeri is really restricted to Southern California. Okay, so to finish up the talk, one of the things that I think we should be doing moving forward, at least in this field, is to try to link these environmental drivers of transitions in sociality with the genetic drivers. And because we're just starting to find these genetic drivers, these links are still sort of up and coming. So one of the things that I want to do going forward is to try to distinguish between whether this transition is due to ecological factors and then becomes canalized, forming a genetic basis, or whether this starts off as a genetic selfish gene and then that allows these alternative social forms to colonize different environments. So the idea here, I know it's kind of a confusing diagram, but if we have a monogenous ancestor living in one environment represented by the palm tree, and then it colonizes a novel environment and suddenly polygyny is favored, this can lead to the canalization and ultimate genetic basis of polygyny. Alternatively, with the genetic route, we might have a mutation leading to the origin of a selfish gene, and then phenotypes associated with the selfish gene can allow expansion into novel environments. So these are very hard to disentangle, but I think the way forward is to use a comparative approach. So try to get down to the basis of, hopefully we have some suggestion that there may be basal Formica that are socially polymorphic but lack the supergene, and I think that that's where we can start to try to disentangle these two different factors. So take-home messages, there's a really cool ancient supergene in Formica, a lot of interesting stuff to do with it. We also have gradients in queen number along environmental axes. How do we put those together? That's the next step. And we have lots of other stuff going on in the lab, and my graduate students are dragging me in all kinds of new and exciting directions too. But thank you for your attention. I'm happy to answer questions.
You might not know the answer to this. This is just kind of, I like a fun fact. You're talking about how the fire ants, I think my question is exactly what I think it's right. You're talking about how the fire ants know there's a double B. No, here's my question. How do worker ants know that the queens are a BB and kill them? Like, do they have a tattoo? Like, how do
Yeah, they smell different, exactly. So they think that there's an odor difference. It has been somewhat quantified, but not as carefully as it might be. But yeah, there's an olfactory signal. Yep.
Along those lines, so you mentioned that the eggs don't develop them to larvae.
Right.
First of all, just a clarification, was that done in isolation, or were the eggs?
In isolation,
so
yes. So we had two treatments. The one that I showed was in isolation. We also had them being tended by all manner of workers, and in no case do they ever develop. So we think it is an intrinsic mechanism.
Right, so it's not necessarily the organism. So then
correct.
What kind of intrinsic mechanisms do you think are happening?
So the one that's the best described is this Medea system in the flower beetles, where essentially the mother transmits a toxin in the cytoplasm, and the antidote for the toxin is contained within one haplotype, and so they have to also receive that haplotype. So we're looking for a signature of that. I mean, I have to say that so this is work that's being done in Michel Chapuiset's lab, and nobody there is really molecular enough to get at that as well as one would hope. So we're hoping to find collaborators who can help us kind of get all the way there with the mechanism. But that would be my prediction. Other questions? Yep.
So I think that you only see this when the queen is heterozygous, right?
That's right, yeah. So if we can cross PP queens with an M male and they only produce heterozygotes and they're fine. Yeah.
So I know some of the ants with the multiple chromosomes are like invasive.
Correct.
And the ratios are skewed more when they invade, or often towards the multiple. And I know that some of those like fire ants aren't kind of the populations that invade tend to be questioned elsewhere.
Right.
And in part it's linked to passing to a bottleneck and losing variation on invasion. I'm just wondering, have you looked at effective population size? Like, do you find that that's sloughing off of diversity as you go higher in elevation? Do you think there's some?
That's a great question. Yeah, so that's a great question. We're still investigating whether there could be a sourcing dynamic. So one of the elements of the polygyne syndrome that hasn't really been well confirmed in any Formica species is that the monogynes should be better dispersers because the queens tend to be larger. They have more fat reserves and they might be more able to disperse further and found nest independently. So one possibility, and than the multiple queen colonies, or the queens from multiple queen colonies. I'm getting myself tangled up. Anyway, these better dispersers might be more likely to just get up to this marginal sink habitat at higher elevations, resulting in the same pattern that we see. So we have a large-scale population genetic analysis going on now. So this is with data that I collected being led by a graduate student in Lausanne. And we're not really finding evidence of that. So we're not finding private alleles at higher elevation. We're not finding eroded diversity at higher elevation. One thing that is interesting, so there are also differences in the effective queen number of polygynous colonies at higher elevation. So we do find that the polymorphism is retained across all of these different populations, but the polygynous colonies have fewer queens, and the queens, this is very anecdotal, seem to be more heterozygote. So there's something interesting going on, but I think if you'd asked me a couple of years ago, I would have said source sink. Now I think it might be some sort of selection going on. Yes?
So it sounds a lot like the evolution of the P element in Drosophila.
Yes.
But these differences, there's lots of P elements transposed, but yours are restricted. So the question I would raise is, what kind of biochemical gene difference can you imagine have to be dominant? And in the case of the sex supergene, it was obvious.
Right.
It was biased towards one sex. So what biology are you biasing?
Yeah, that's a great question. I mean, we're still in the early stages of actually looking at the genes underlying this variation. I mean, our best candidate at the moment is a transcription factor, which is both promising and kind of discouraging because we're not going to have an instant answer. So we need to look at the interacting gene networks with that transcription factor and figure out what's going on. But yeah, I mean, the short answer is I don't know yet, and I would love to talk with you more if you have ideas for first steps on how to look at that.
Well, another candidate would be SRY, essentially a poison.
Yeah, I mean, that's right. So the gene that fits best within that very old strata that I highlighted, so that one little region that's conserved across 15 genes, is called, inconveniently, called knockout in Drosophila. So try Googling knockout gene. So it's called knockout. It's a transcription factor, but it also arrests development during. So it's implicated in early neural development in Drosophila. So it's possible that is breaking down in those early stages of development. And what we're trying to do now is track its expression through different genotypes across the developmental trajectory. Of Formica, we had an inconvenient year because so much rain came late, so we're not finding a lot of brood. But I mean, that's sort of our best guess at the moment. But whether that's actually the selfish gene in Formica, I'm actually having doubts now because we're finding that the function seems to be different in different species. And so I'm not sure that the way that the selfish gene works, that maternal effect killer, is conserved. It's just that this region has something going on that's conserved.
Are you able to do essentially quantitative transcription assays to find out what kind of RNAs or proteins that might give you some idea of biology?
That's right, yeah. So the lab where I did my postdoc, the postdoc who came after me started that project and then left science. So we're kind of in this awkward moment. But I heard recently from Michelle that somebody's picking it up with Formica selysi, which is the optimal one to work on at first because we know the most about it. And my goal is to try to expand this into some of these North American species that are readily available to me, but also may have some interesting other stuff going on. But yeah, it's coming. Stay tuned. Yes?
So one of the things with the polygyne species is that they're very polygyne. So there seems to be sort of a dearth of species that have, say, 4 or 5 queens, or some low number. So either you have a single queen or you have many queens. And the argument against the polygyne is that they just sort of lost the ability
Yeah. So in Formica is not so extreme. I mean, fire ants are completely, I agree, they're like 200, 300 queens is totally common. In Formica, we've never found more than 15 in one nest. And we do have a really rare and weird intermediate that we call oligogyne, where there are two, we think, highly related queens that are functionally monogynous. So like they have the MM genotype. And so it is a nice system to sort of work it, to look at the regulation of queen number and sort of acceptance, because there do seem to be limits in this species. It may also just be that the populations of the species, because it's the main floodplain specialist in the area, they're so saturated that, I mean, polygynous queens have their choice of colonies to join. So it's not clear to me whether it's sort of an ecological constraint or whether there's something intrinsic that's saying, okay, we have enough queens, don't take any more. But, and it's possible that we're not seeing many of them, but on the cool spring day, they all seem to be, we put down these nice flat stones in the field so we can open them up and all the queens tend to be right there. So it's nice. It's a nice system.
So is it as simple, can't be as simple as the poly kind of females are maybe smaller, and if they tried to make it alone, their colony wouldn't make it?
They are smaller. We can...
But can you make them larger? If you isolated them and fatten them up or something, would they
right.
take on the appearance of monogeny?
So the grand we, so a lot of graduate students in the lab, not so much me, have done lots of independent founding experiments. They've found no difference in the lab, under lab conditions, in the ability of queens of polygynous origin and queens of monogynous origin to found independently. Both can produce nenitic workers. So these are the tiny workers that they produce by digesting their flight muscles initially. And so both can survive for multiple years in this lab setting. We can provide food supplementation. It has a similar effect in both. So they generally won't eat until they've produced nenitic workers, whether or not they have food available. And then they'll start to pig out and quickly increase their reproductive rate.
So it's unlikely that there's a gene called polygyne that it says that's what I am. But just in terms of the balance and biases,
right.
And maybe what you're looking at is the loss of a anti-gynous polygynous system.
So the loss of something that would cause individuals to to discriminate.
Females can't stand each other.
Right. Yeah, I mean, I think that there is something like that going on. So we haven't done any. So people have done queen introduction experiments and they haven't found a lot of discrimination by workers. So we've tried the same things that result in this very strong discriminatory response in fire ants. That doesn't seem to be the case in Formica. So there isn't, at least among workers, a strong response. The queens don't seem to have much of a say in who gets into the colony. The workers choose, at least in the experiments that I know of. But yeah, I mean, it's entirely possible. And I mean, I do think that there is something of...
So are you suggesting that this is the phenotype is actually manifested by the workers, not by the queens?
Indeed, at least that's the conventional wisdom on, or the current wisdom on sort of who are the gatekeepers for the colony. So if you have a new queen that comes to join, the workers are the ones that are deciding yay or nay, not the queen.
I'm going to stop this presentation offline.
Thanks.